presented by Toshio Kota from University of Alberta. And he is going to be presenting his strategy of using antisense gapmers for the treatment of FSHD. So thank you and welcome.
Thank you, Beth, for introduction. And I'd like to thank Dr. Beth Barton and organizer for having me here today at this very exciting meeting. So my name is Toshio Kota, and I'm professor at the University of Alberta, and I'll talk about development of antisense gapmers for the treatment of facioscapulohumeral muscular dystrophy, or FSHD. Okay. Yeah. Okay. Great. So this slide is a bit of introduction. So our main focus is antisense oligonucleotide mediated therapy, and in short, this approach is to fight diseases using synthetic single-stranded DNA-like molecules. It's called antisense oligonucleotide targeting RNA. So here, synthetic is the keyword. So for antisense therapy, antisense oligonucleotide therapy, we always use synthetic chemistry. So that makes a difference from other similar approach like siRNA or RNAi. And these are some examples of synthetic antisense oligonucleotides. Okay, so yeah, so there are a couple of different approaches of antisense therapy. So one way is to knock down the gene expression, and another one is we call knock up, or to restore the gene expression. So we can knock down gene expression using a couple of different approaches. One is it's called steric blocking. So you can block translation or transcription of DNA or RNA. But recently developed method is called gapmers. Gapmers is much more powerful way to knock down gene expression. And basically you can use chimeric antisense oligo called gapmers. So central part is DNA, and both ends are modified antisense oligos like LNA or 3' MOE, and this central part induce RNSH, and it leads to knockdown, degradation of RNA. So knock-up approach is a bit of different approach. So basically we can use antisense oligo for splice switching. So we can either inhibit splicing or stimulate splicing using antisense oligos to skip exon or include exon, and it can restore gene expression. This is a knock-up approach. So in the next slide, this is a bit of history of antisense therapy. So the overall concept of antisense therapy was discovered, developed in 1970s, and gapmers and splice switching antisense oligos all developed in 1980s and 90s. So it's not a very new approach, but these gapmers and splice switching oligos are really used quite recently for rare diseases. And from 2013, I think golden era of antisense therapy started. So to date, almost 10 antisense oligos were developed to treat rare diseases. And many of them are for neuromuscular diseases. And these are all either gapmers or splice switching antisense oligos. So I think it highlights the importance of these technologies. Okay, so yeah, and this slide is our previous study. So in 2009, actually our study was first study to show restoration of dystrophin by exon skipping in a severe DMD animal model, which is dog model of DMD, and it prompted collaboration with Japanese pharmaceutical company Nippon Shinyaku. And after lots of preclinical work and clinical trials, it's approved by the FDA last year. So we are very excited about this. And it took 10 years, but we got there. And so we are in particular very excited by the approval of Butolarsen. And it's now available in several countries. So for so facioscapulohumeral muscular dystrophy, we use gapmer approach. So basically FSHD is autosomal dominant disease, and 8,000 to 22,000 are affected. It's progressive spreading muscle weakness and rare infantile form also exists, and this is a much more severe disease. So this is basically the third most common form of muscular dystrophy. And the this disease, FSHD, is caused by misexpression of DAX4. DAX4 is a very toxic gene. It's usually expressed only in very early stage in development, and it's not expressed in adult. But in FSHD patients, it's misexpressed in muscle, and it's only expressed in healthy testis in healthy individuals, but it's very toxic in muscle. So we target this gene basically using antisense oligos. So here we use a couple of different models. One model is FSHD patient-derived myotubes, and this is immortalized cell developed by French group and UMass group. And another model is FSHD inducible mouse model. This is developed by University of Nevada group, and we test our antisense oligos in these models, and we looked at the effect of DAX4 knockdown in these models. Okay, so this is a design of antisense oligo gapmers. So basically, we mostly target exon 3 in DAX4 gene because all toxic form of DAX4 have this exon 3. So our main target is exon 3, and we use cell model. So the thing is DAX4 is only expressed in very mature myotubes. So the detection of DAX4 in myotube is very difficult, even in patient-derived cells. So we differentiate for almost 2 weeks to look at the effect of our antisense oligos. Okay, so our antisense oligos worked very well. Some of them induce more than 99% knockdown. We use 2 different types of antisense chemistries. One is RNA gapmer, and the other is 2' MOE gapmer, and they both work very well, equally well. We can induce more than 99% knockdown at 100 nanomolar, very low doses. So this is very encouraging. And so this is dose dependence. So we tried 3 different doses, and it can induce significant knockdown at 10 nanomolar. So this is very exciting. And we also looked at downstream gene. So there are several downstream genes known for DAX4, and our antisense oligos, basically almost all antisense oligos also significantly reduce the downstream gene. So this is very exciting as well. And we also looked at RNA-seq. So basically, what we wanted to do is we look at the overall change of the RNA expression pattern, profile of RNA expression in patient cells, and if this effect can be reversed to normal levels after the treatment. So our study, and to offset the noise, we used previous studies to identify upregulated genes and downregulated genes in FSHD. So we call them FSHD signature genes. Okay, so this is a bit complicated slide, but so what this slide want to say is, so before treatment in non-treated FSHD patients, so some genes, many genes are upregulated. These are all red dot, so right side in the slide. And some genes are downregulated. These are blue dots in this slide. And after the treatment with antisense oligos, these dots shift to center. So that means expression profile of these signature genes become shift toward normal level, normal state. So this overall highlight that antisense oligos can return the state of cells towards normal level. Okay, so also we looked at the morphology of the cell, and so in FSHD patient cell, they have problem in muscle differentiation. They have issue in muscle fusion. So compared to untreated healthy cell, non-treated FSHD cell, they don't fuse very well. They are still myoblast and mononucleated cell, and they are much more thinner compared to unaffected healthy cells. And after the treatment, for example, if you look at LNA6, they form more mature myotubes and thicker compared to untreated one. And so we can see significant difference of the thickness of the myotubes or fusion index of the myotubes. So after the treatment, apparently myotubes differentiate better. So this is also encouraging result. Okay, so this is statistics, and so basically cells diameters are increased after the treatment, and after the both LNA gapmer and 2' more gapmers. So we are also looking at the effect in mouse model of FSHD. It's still ongoing in Dr. Ewen Chen's lab in Washington, D.C. So basically, we have inducible mouse model, but because DAX4 is very, very toxic gene, even without inducing DAX4, they have very small amount of leaky expression, and it can efficiently induce FSHD muscular dystrophy. So it turned out to be a very good model. And it's developed by Dr. Peter and Takako Jones' lab in University of Nevada. So after the intramuscular injection, we can, so this is fluorescence labeled LNAs and gapmers, and we can see gapmers really have its uptake in muscle fibers. And also we can significantly reduce the expression of DAX4 gene using LNA gapmer and 2' memory gapmer. So far, both LNA gapmers and 2' memory gapmers work quite well, and they are both quite promising. Okay, so this is summary. So we designed 2' memory gapmer and LNA gapmers, and they strongly knock down DAX4 expression both in vitro and in vivo. And these gapmers also replace expression of signature genes and downstream genes in vitro. And these gapmers improve the cellular phenotypes of FSHD myotubes. And we are also working on systemic treatment of gapmers in FSHD mouse model. Next slide. Yeah, this is contributors. So this study basically done by Kenji, my PhD student, and these are our collaborators. And these are funding